Welcome to STEM Pathway. Today we are opening up the human body. We are going to have a live surgery right here in the school laboratory to map out the hidden cavities that hold life together, right from the pump of the heart to the core of the gut. We are dissecting it all layer by layer. We're going to look at different cavities, and in this case, we're going to look at the parts of the human anatomy, right from the brain, the human heart, the liver, which filters toxins, then the stomach, the large intestine, the small intestines, and even the urinary system. So in this case, before we proceed with our surgery, we are going to use very simple tools in this laboratory. But in this case, we're going to begin by looking at the model on what we are going to expect in this our surgery. So in this case, we're going to have some parts which are visible here, like you can see. We have the kidneys, like in this case, we have the right and the left kidney. Then we're going to have some blood vessels, like you can see here, and also the urinary system, where we are going to look at what is found there. Then after that, we are going to look at the parts that we are going to extract from our surgery. Like in this case, we're going to have the small intestine with the large intestine, and then we're going to have the liver, like you can see it here. Then we're going to have the stomach below the liver, and then we also have the heart, or the human heart, with these two arteries. We have an artery and then the aorta. Then we're going to have another important part which receives oxygen when we breathe, and that is the lungs. And in this case, we're going to have these right lungs, and then we're going to also to have this left lung. And then after that, we're going to rearrange. After the surgery, we're going to rearrange the body to be in this format. Before we begin, we are going to have the simple tools that we're going to use. And one of the simple tools that we're going to use in this case is this stethoscope, which we're going to use to listen to the heartbeat of this specimen. And in this case, this stethoscope is going to be very instrumental in this case. Then after looking at this simple tool which we're going to use, which is the stethoscope, then we're going to look at this model, which is going to guide us on how we are going to remove or rearrange these particles, all these parts of this human body. The first thing that we must extract is the lungs, as you can see it here. This is the left lung, and then we also have the right lungs, like that. That is what you can see here on the screen. Then we're going to have the heart, which you can see it here. That is the third thing that we're going to extract. So be very keen on how we will extract it, so that we don't mess up with the live organs. Then we're going to have this liver, as you can see it. So we're going to remove also this one as the fourth part. Then we also remove the stomach. That is the stomach that you can see here, which has just fallen, sorry. That is the stomach. We're going to remove as the fifth part of this model. Then we're going to have now the small intestine surrounded by the large intestine, as you can see there. So that is the intestines, where we'll have the large part of it being the small intestine, and then we have the blue or the grayish large intestine. So that is the sixth thing that you're going to remove. Then after that, we are going to have these simple and simple nodes, and these simple nodes in our case here are going to have some cards, and these cards are going to have instructions on the step-by-step what you're going to do in our simple surgery in the school laboratory. So here I want you to be very keen on how we are going to arrange these cards, and these cards should be arranged in an order in which we are going to extract the parts of this specimen, so that we don't mess with the specimen. Remember, when we are doing a surgery or when we are handling specimens, we should not tamper with the life of the specimen. So these cards are going to give us the instructions on exactly what and where we are going to touch for us to not mess up with the specimen. And you can see the cards here. These are the cards. So be very keen and be very attentive as we move forward now back into our school laboratory. And this one might be the first experiment to be done successfully in a school laboratory, where we are going to handle a live specimen right from the beginning to the end of the experiment, and the specimen will remain alive, as we are going to see in the next few minutes. So this is the model that we're going to use, and this is the notes that we're going to use in this process. Now, let's get started.